Hi everyone, so I'll share my screen and we can start our session for today. Okay. Okay, so there's some issues, so I think I have, yeah, so I have shared my screen. Can you guys see the screen? Can one of you unmute and let me know if you can see the screen? Can one of you let me know? Yes. Yeah. Okay. Yeah. Okay. Thank you. So, welcome to the sixth week of these live sessions. So, this is a live session based on the course Genetic Engineering Theory and Applications, taken by course instructor Professor Vishal Trivedi from IIT Guwahati. And in these live sessions, what we do is that we discuss some sample problems from sample assignment problems from last year, and we try to figure out the solution to those problems, right? And we try to discuss the concepts behind those solutions so that it's easier for us to solve similar type of other questions, right? And we want the sessions to be as interactive as possible. So if you're not able to understand anything or if you have any doubt, please don't hesitate. Either you write in the chat box or you unmute and let me know. And if I'm able, I know the answer, I'll answer now. If I don't know, I'll read about it and let you know by the next class. Okay. So with that, we'll start the session for today. So my name is Aishi and I'm doing my PhD in the Department of Chemistry, IIT Bombay. And we'll start today's session. So the first question for today is: degradation of pathogenic bacteria inside a cell occurs in the secretory vesicle, phagolysosome, RER is a rough endoplasmic reticulum, and SER is a smooth endoplasmic reticulum. So which one do you think is the correct answer? I'll wait for 2-3 minutes, then we'll move on. Anybody?
Ma'am, the answer would be phagolysosome.
Correct. Anybody else has any different? Sorry. Anybody else has any different opinion? So yes, the answer will be 1 second. Yeah, the answer is phagolysosome. So anything foreign that comes from outside uh, basically gets engulfed by the phagosomes, and ultimately the phagolysosome takes care of it. So we'll move on and talk about the entire process. But what is phagolysosome in brief? So phagolysosome or an endolysosome basically is a cytoplasmic body formed by the fusion of a phagosome with a lysosome, and in a process that occurs during phagocytosis, right? So formation of phagolysosomes is essential for the intracellular destruction of microorganisms and pathogens, and it takes place when the phagosomes and lysosomes membranes collide, at which point the lysosomal contents, including the hydrolytic enzymes, are discharged into the phagosome in an explosive manner, and they digest the particles that the phagosome had ingested. So some products of the digestion are useful materials and are moved into the cytoplasm. Others are basically exported by the exocytosis, right? So membrane fusion of the phagosome and lysosome is known to be regulated by various proteins, out of which RAB-GTPases is one. Okay, which basically allows the exchange of material between these two organelles, but prevents the complete fusion of their membranes, right? So if we talk about the immune system, basically we need to start from a basics, right? So the immune system and its importance for homeostasis of an organism. Homeostasis is basically the normal condition, right? How we are able to maintain a normal condition up till a certain point. Correct. So the immune system is a system of non-specific and specific mechanisms that protect us from damage caused by infectious factors of the environment, and providing surveillance of its own structures, like any kind of elimination of damaged or dead cells and all of that. Now the aim is the maintenance of homeostasis and the integrity of macroorganism, right? So what is an antigen? Antigen is basically a substance foreign to us that is recognized by the immune system, and finally when the immune system reacts to it. So it can originate from environment, so it will be called an exoantigen, or it can originate from our own structures, autoantigen. So if our own body immune system recognizes our own bodily substances as autoantigens, that can lead to a disaster, because that can lead to autoimmune disorders. I think you have heard about various autoimmune disorders, some being rheumatoid arthritis and all of that. So the basic uh, concept of autoimmune disorders is that your body immune system is not able to differentiate between self and non-self things. So it is reacting against its own bodily things. That can actually be quite disastrous. Now, usually proteins or polysaccharides like lipids or nucleic acids only combined with proteins or polysaccharides can also act as antigens. Now, what are the mechanisms of the immune system and their cooperation? The innate immune system. So the innate, it's not developed after the exposition to infection, and it has a uniform response, like prompt, no immunological memory, right? It can be the mechanical barriers like mucosa, skin, phagocytic cells like macrophages, and the acute phase proteins like the C-reactive protein, the complement system, and all of that. Correct. Then we have the adaptive or the specific immune system. So the innate immune system is basically the first line of defense, right? So it's not that specific. Anything suddenly attacks you, you need some non-specific warriors to fight for you. Then there comes the adaptive or the specific immune system. So here the key role is adaptability, developed after the exposition to infection, and the immune system is not inherited. Immunological response and B and T cells like the immunoglobulins. So what are the components of the immune system? The innate immune system will be neutrophils, eosinophils, basophils, macrophages, natural killer cells, NK cells, etc. And then you have the adaptive immune system like lymphocytes. Now, what are the organs? Liver, skin, etc. And there can be primary lymphoid organs like bone marrow, thymus, all of that. And there can be secondary lymphoid organs like spleen, lymph nodes, mucous, acidic lymphoid tissue like mold, etc. Right? They can be what are the substances of the immune system, the complement system. Cytokines, like molecules which ensure the communication between the components of the immune system, acute phase proteins, then hormones, immunoglobulins, etc. Right? Now, what is the immune system of the mucosa and skin? The mucosa is an intact surface, clearance of surface like lysozyme, IgA, lymphoid tissue of mucosa, and macrophages. Skin will be an intact surface. There is a lipid barrier, eczema, and then immunocytes of the skin, like the Langerhans, the Langerhans cells. Now, what are the non-specific immune mechanisms? As I said, barriers like skin, mucosa, or there can be secretions like fatty acids, hydrochloric acid in the stomach, lysozyme, all of these. And various physiological mechanisms like body temperature, hormones can also act as these non-specific immune barriers. And then phagocytosis. So, what is the process of phagocytosis? The process by which particular substances or cells are ingested and destroyed by specialized cells. Like neutrophils, macrophages, they can do phagocytosis. So, what happens basically? There is diapetesis, chemotaxis, ingestion, phagosome, phagolysosome, industrial destruction. So we'll talk about all these in some the upcoming slides. Okay. So as I said, the complement system consists of at least 20 serum proteins, which once activated acts like a part of the innate immune system. And the complement components are present in serum in inactive form, and the complement is activated in a cascading manner. So complement proteins are synthesized mainly in the liver, but tissue macrophages and fibroblasts can synthesize some complement proteins as well, right? So the complement protein system has there are three paths of the complement activation. Classical, they are activated by the complex of an antigen and antibody. Then alternative, activated by reaction of C3b with foreign substances like liposaccharides of distinct microbes. And then lectin, that is activated by binding of the MBL to the microbial surface. Okay, so we'll not go into the huge details of the complement system, just what are the basic functions. Inflammation like degranulation of mast cells, chemotaxis, increased vascular permeability, diapetesis, activation of polymorphonuclears, NK cells, and macrophages. Then clearance of immunocomplexes, lysis of the cells like G bacteria, protozoa, neutralization of viruses, and then opsonization. Okay, so what is opsonization? We'll also discuss all of this. So how is the complement system regulated? So some serum proteins enzymatically attack complement components and they can inactivate them. Some serum proteins can bind to and thus inhibit complement components, right? And then there are some regulatory proteins in the cell membranes that can act in its favor. So the immunological mechanisms of inflammation, what is a local? So the activation of hemocoagulation, then synthesis of kinins like bradykinin, vasodilation, vascular permeability, edema and pain. Then cell-mediated response, acute, chronic, tumor, etc. Healing, restoration of tissue architecture, scar tissue development, all of these. Okay. So what are adaptive immune mechanisms? One can be humoral, generation of antibodies, IG, by B cells, plasma cells. In majority of antigens, the cooperation with T helper cells is necessary. Then it can be cell mediated, like generation of antigen specific T cells, like helper or cytotoxic. Antigen presentation is necessary. Now, adaptive immune mechanisms can be antibodies, neutralization and opsonization of like microbes, complement system activation. T cells can be cytotoxic effects to microbes, help for B cells, macrophages, activation cytokines, etc. Now, the innate or non-specific immune system can prompt reaction in less effective, less directed. The adaptive or the specific immune system can help in slower development of reaction, more effective, more directed immunological memory. And both systems can cooperate. Complement system is activated by IC. Cytokines recruit other cells to the site of the reaction, and then antigen presentation. So daunting role of the immune system is to basically afford protection, right? So it is designed to recognize and respond to non-self antigen in a coordinated manner. So additionally, cells that are disease, damaged, and distressed or dying are recognized and eliminated by the immune system. Right? So to accomplish this goal, the immune system is equipped with a rapid response mechanism, exquisite specificity, adaptability, and intricate regulatory network and memory. Now, the immune response generated against a potential pathogen is called, so the response is called the immune response, right? And it can have two complementary arms. We have already discussed innate and acquired immunity. Now, innate is the immediate response. Immediate response when the pathogen attacks you, it represents the first line of defense, which is not directed against any particular pathogen, but a general defense mechanism. So it is non-specific. It is active right from the time a child is born and is rapidly mobilized at the initial site of infection, but lacks any kind of memory. Like if the same pathogen attacks again, this will again show a non-specific uh, defense, and it includes barriers to the infectious areas like skin, our epithelium, and then mucous membranes. All of these fall under innate immunity, and it also includes many immune components important in the adaptive response, including phagocytic cells, natural killer cells, TLR, the toll-like receptors, cytokines, and complements. So we already talked about mechanical and physical barriers. These are which help in innate immunity. Skin, obviously, few organisms are capable of penetrating intact skin, but many can enter sweat or sebaceous glands and hair follicles and establish themselves there. Now, the sweat, whatever happens when we are sweating, so even our sweat is important because it has antimicrobial properties like acidic pH. So, not all microorganisms can survive, and then certain chemical substances like fatty acids. Then we come to lysozymes. Lysozyme is a hydrolytic enzyme present in all mucous secretions, including our tears, saliva, skin, in respiratory and cervical secretions as well, that can lyse gram positive by cleaving peptide or alkaline layer found in bacterial cell wall. So you see, even when you're crying, your tears are acting as as a first line of defense. Your tears have lysozyme, which can actually lyse gram positive cell wall. Then we have psoriasin, which is a protein with antibacterial properties produced by skin. Now, the mucous membranes in respiratory tract, they tend to, in case of bacteria, they tend to stick to a film of mucus covers the surface, and it's constantly being driven upward by ciliated cells towards the natural orifices. We have lysozymes, we have immunoglobulin A, we have phagocytes, and we have hairs at the nose and the cough reflex. So that is also a special protective mechanism. Then we have the gastrointestinal tract. The saliva contains numerous hydrolytic enzymes, acidity of the stomach, proteolytic enzymes, and active gives in small intestine. Right? Now, a constant normal microbiota that itself opposes establishment of pathogenic microorganisms, that is called bacterial interference, and has important physiological conditions. Example, in the vagina, the acidic pH is maintained by normal lactobacilli. So it's a bacteria, but it's helping you maintain your acidic pH so as to stop any other pathogenic bacteria from coming. Okay. So it inhibits the establishment of yeast infection, anaerobes and gram-negative bacteria. So we have talked about that it has both cellular and humoral responses. Phagocytic leukocytes such as PMN polymorphonucleosides, and then macrophages, NK cells, etc. Now, neutrophils, the mechanism of neutrophils is basically phagocytosis, basically engulfing foreign pathogen, and intracellular killing, inflammation, and tissue damage. For macrophages, it's phagocytosis and intracellular killing. Extracellular killing of infected or altered cell targets, tissue repair, and then antigen presentation for specific immune response. For natural killer cells, it's killing of virus infected and altered cell targets, and then for eosinophils, it's killing of certain parasites. Now, the interaction of the invading microbes with these cells and other cells throughout the body triggers the release of complements, cytokines, and chemokines. So, armed with these tools, the host initiates its defense mechanism against the invading pathogens. Now, what are the humoral components? Complement, as I said, it helps in lysis of bacteria and some viruses, opsonin, increase in vascular permeability, recruitment and activation of phagocytic cells. Then you have the coagulation system. Basically, it increases the vascular permeability, recruitment of phagocytic cells. B lysine from platelets acts as a cationic detergent. Then you have lactoferrin and transferrin. It competes with bacteria for iron. You have lysozyme breakdowns the bacterial cell walls, the peptidoglycan layer of the gram-positive cell walls, bacterial cell walls, and cytokines, which has various effects. Different cytokines have different upregulatory and downregulatory effects. Now, what are microbial sensors? When a pathogen enters the skin, it is confronted with macrophages and other phagocytic cells possessing microbial sensors. So, what are these sensors? There are some receptors on the surface of these cells which recognize foreign pathogen, foreign antigen. So, it can be toll-like receptors. It will be toll, not tool. Toll-like receptors are TLRs. Not-like receptors are NLRs, and Rig1-like helicases and MDA5. Now, these are pathogen associated molecular patterns. So, the microbial molecules that stimulate innate immunity and present in infectious agents like bacteria and all, these patterns are known as pathogen associated molecular patterns. And then there is PRR, pathogen recognition receptors. So, these receptors of innate immunity are in the host cells that recognize the structures shared by these microbes. Then we have DAMs, damage associated molecular patterns. The innate immune system recognizes molecules that are released from damaged or necrotic host cells. The examples include high mobility group box protein, etc. Now, what are TLRs? The TLRs are a family of evolutionary conserved pattern recognition receptors that recognize the pathogen associated molecular patterns, and they constitute the first line of defense against a variety of pathogens and play a very critical role in initiating the innate immune response. So, till date, 10 human TLRs have been identified, and each receptor appears to be involved in the recognition of a unique set of microbial patterns. For example, TLR recognizes several glycolipids and peptidoglycan that are made by gram-positive bacteria. TLR3 engages double-stranded RNA in a viral replication. TLR1 and 6 recognize multiple diacyl peptides. For example, how we see in mycoplasma. TLR4 is specific for gram-negative lipopolysaccharide LPS. TLR5 recognizes bacterial protein called phagonin. TLR7 and TLR8 interact with single-stranded RNA in viral replication. TLR9 binds to bacterial DNA, and TLR10 remains basically an orphan receptor. That it's nothing has been. People are trying to explore it. Nothing has been confirmed yet. So these are the different TLRs or toll-like receptors on the surface, and these are the various substances that can bind to it. Then you have the node-like receptors, the large family of innate receptors, sense DAMs and PAMs that are located in the cytosol of cells and serve as intercellular sensors for microbial products, right? So what they do is their upcoming job is basically activating the nuclear factor kappa light chain enhancer of activated B cells, NF kappa B pathway, and drive inflammatory responses similar to the TLRs. Now, Rig-like helicases like retinoic acid inducible gene 1 and MDA5, melanoma differentiation associated gene 5. So, these are cytoplasmic sensors of viral single-stranded RNA, and the engagement of ssRNA with these sensors triggers type 1 interferon production, and these interferons are highly effective inhibitors of viral replication. Now, coming back to phagocytosis, during infections, the number of circulating phagocytic cells often increases, and the main functions of phagocytic cells include chemotaxis, migration, ingestion, and microbial killing. So, there are various other exogenous antigens that can enter the lymphatic lung or bloodstream that are engulfed by a variety of phagocytic cells. Now, phagocytosis basically involves extension of pseudopodia to engulf attached material and fusion of the pseudopodia to trap the material in a phagosome, fusion of the phagosome with a lysosome to create phagolysosome, as I was saying. That comes after this, after the fusion trapping of the phagosome. So, fusion of the phagosome with the lysosome will give rise to phagolysosome. Then ultimately, whatever is inside will be digested, and then exocytosis of the digested products will happen. Now, attachment via receptors is phagocytosis. Pseudopod extension, phagosome formation, granule fusion, and phagolysosome formation. All these are steps. So in here you can see the attaching by the receptor. In B there is a pseudopod extension. In C there is a phagosome formation. And D the granule fusion and phagolysosome formation. No, as I said, the phagocytosis process is by which phagocytic cells, especially the PMN, can recognize the pathogens, ingest it, and then destroy the engulfed organisms. This is multi-step process. Like first, the steps of extravasation will include rolling, activation by attractants, arrest and adhesion, transendothelial migration, etc. Now, antimicrobial mechanisms used by phagocytes. So, the acidification occurs within the phagosome. The phagosome pH is 3.5 to 4, and this level of acidity is bacteriostatic or bactericidal. So, toxic oxygen derived products are generated and include superoxide, hydrogen peroxide, and singlet oxygen. There are toxic nitrogen oxides as well. Then there are antimicrobial peptides also that participate in killing. We have talked about this. We have talked about difference between innate and adaptive. The diversity is limited and germline coded in case of innate, whereas the diversity in case of adaptive unit is very huge. Receptors are produced by somatic combination of gene segments. There is no memory for innate immunity. That means if you are attacked by the same pathogen again, they will not recognize that as pathogen. There is memory system for adaptive immunity. And non-reactivity to self, also innate immunity has a way of reacting with self antigen, which is very harmful. Adaptive immunity also has kind of this affinity. Now then these are the components. What are the chemical and cellular components for adaptive immunity? The lymphocytes in epithelia, antibodies secreted at the epithelial surfaces. Blood proteins will be antibodies, okay, like complement was for innate, and cells will be lymphocytes. So you talked about the defense of early infectious microbes, the early reactions and the late responses. So you can see from here it's a basically a graph. So on the x-axis we have our time after infection. So you see how these structures come into say like in the time after infection in the first hour we see the first to 12th hour we see the innate immunity doing its work like epithelial barriers, phagocytosis, complement proteins activation, complement cascade activation, etc., natural killers, etc. Then from 12 hours onward to 7 days or 10 days we 7 days or 10 days we see the adaptive immunity coming into role. The B lymphocytes giving rise to antibodies, the T lymphocytes giving rise to effector T cells, etc. Now, in case of innate immunity, as I said, there are functions and components and their influence on the adaptive immunity. So, this is an evolutionary older defense strategy mechanism found in all classes of plants and animals. Pre-exists infection or exposure to foreign antigen. It responds immediately after recognition of pathogens like microbes, and it responds in essentially the same way to repeated infection. Okay, that's because it is innate immunity. So the initial response to microbes to prevent infection with low elimination of danger. Innate components are also important in the adaptive immunity, stimulates adaptive response. Okay, so we are seeing what are the epithelial layers, defensins, and all of this their function, microbial killing, prevent microbial entry, and the circulating effectors such like neutrophils, they do early phagocytosis and killing of microbes. Then macrophages basically they do efficient phagocytosis and killing of microbes, secretion of cytokines that stimulate inflammation. Then we have NK cells that lysis of infected cells, activation of macrophages. Okay, then we have the complement system, killing of microbes, opsonization of microbes, activation of leukocytes. Then we have the MBL, mannose binding lectins, opsonization of microbes, activation of complements, etc. We have C reactive proteins, opsonization of microbes, activation of complement, etc. So you see another schematic of the different epithelial barriers, physical barrier to infection. Then we have the killing of microbes of any non-locally produced antibiotics, killing of microbes and infected cells by intraepithelial lymphocytes. They are the routes for infection for pathogens. For airway, we have the inhaled droplets. For gastrointestinal tract, we have the contamination with food or water, basically food poisoning. Physical contact can also be one of the reasons. Minor skin abrasions like puncture wounds, handling infected animals, all of these can lead to pathogenesis. Insect bites can sometimes also lead to pathogenesis. We have talked about this. So, what are the antimicrobial peptides? Defensing these are cysteine rich peptides with 29 to 34 amino acids. They are present in the skin and abundant in neutrophil granules. They are broad spectrum antibiotics upregulated by inflammatory cytokines. Then we have the cryptocidins secreted by the epithelium of the intestine and located stimuli by the lumen of intestine. We have the intraepithelial cells, T cells, present in the epidermis of the cell and mucosal epithelia, and they serve as sentinels at common sites of microbial infection. Then we have B1 cells present in perineal cavity, produce natural antibodies. So we read this cell types and primary functions we identify, ingest, and destroy microbes. So this is polymononuclear neutrophils. Okay, PMNs. So these are the most abundant population of circulating leukocytes. They mediate the earliest phase of inflammatory responses. The macrophages have a dominant effector cells of the later stages of the innate immunity response. They play central role in both innate and adaptive immune responses. Sets the functional response of pathogens is active recruitment of the cells to sites of infection, recognition of microbes, phagocytosis, destruction of ingested microbes. Okay, so you can see the microbes bind to the phagocyte receptors. Okay, and then the phagocyte membrane zips up around the microbe. The microbe ingested in the phagosome, then activation of the phagocyte, then killing of microbes by lysosomal enzymes in phagolysosome, killing of phagocytosed microbes by ROS, reactive oxygen species, and nitric oxide. Now, what about the functions of NK cells? This is a type of toxic lymphocytes. Their role is defense against infections by viruses and other intracellular microbes. It rejects tumors also. So the mechanism of effective function has been shown over here. Okay, so there is a virus infected cell that gives rise to stress cell, and that ultimately moves ahead with the effective function. These are microbes, macrophages with phagocytosed microbes. Talked about this. We talked about complement pathway, classical alternative and lectin pathway. We have talked about all this. The complement activation will recruit inflammatory cells, oxidation of pathogens, and killing of pathogens. Okay, then we have cytokines. Cytokines basically are proteins secreted by the cells of innate and adaptive immune response that mediate many of the functions of these cells. Cell source can be macrophages, lymphocytes, neutrophils, etc. And their function is to recruit and activate leukocytes, right? Reduce systemic alterations that contribute to the potential antimicrobial responses. You see, activated macrophages can actually secrete a range of cytokines. They can activate vascular endothelium, activate lymphocytes, local tissue destruction, increases excess of effector cells. Then we have antibodies vascular endothelium and increases vascular permeability, which tends to increase entry of IgG. These are cytokines, and they will, like, some are involved in inflammation, some in control of inflammation, some in interferon gamma production and all. As a summary, the first line of host defense is microbes innate immunity. Mechanism of innate immunity exists between exposure to microbes. The components of the innate immune system include epithelial barriers, leukocytes, circulating effector producers like cytokines, etc. And neutrophils and macrophages are phagocytes that kill intestinal microbacteria, providing producing a reactive oxygen species and all of that. And we have NK cells that are lymphocytes that defend against intercellular microbes by killing infected cells and providing a source of macrophage activation, cytokine interferon gamma. Complement system is activated by microbes and products of complement activation promote phagocytosis and killing of microbes and stimulate infection. So different cytokines of innate immunity recruit and activate leukocytes, enhance the microbial activities of phagocytes, and stimulate NK cells and T cell responses. So what are the chemical factors? These are the biological factors like squamous epithelium, normal microflora. Chemical factors will be sweat, HCl, tears, saliva, defenses, surfactants, etc. For the humoral components, you have the complement, as I said, lysis of bacteria and some viruses, opsonine, increase in vascular permeability, recruitment and activation of phagocytic cells. For coagulating system, we have increased vascular permeability, recruitment of phagocytic cells, beta lysine from platelets, cationic detergent. So all of these will give rise to antibacterial to bowel system. Acute phase protein is antibacterial. Lysozyme breaks down bacterial cell walls, peptidoglycan layer. Lactoferrin and transferrin compete with bacteria for iron. And cytokines have various effects. Interferons are antiviral proteins. So the acute phase proteins are a large group of plasma membrane proteins, plasma proteins whose concentration increases during inflammation, that is injury and in disease states. So pro-inflammatory cytokines stimulate the hepatocytes in the liver to synthesize and secrete acute phase proteins. So examples of acute phase proteins are CRP, MBP, haptoglobin, globin, etc. And CRP binds to membrane phagosolipids in microbial membranes. So it binds to mannose sugars found in bacteria and fungi. These functions as opsonins, soluble pathogenic receptors, activate the complement pathway or be involved in sequestration of essential nutrients. We have the antimicrobial peptides that form pores in cytoplasmic membrane and a variety of bacteria, causing leakage of cellular needs. We have the lysozyme that I already mentioned. We have the beta defensins, are short peptides found in body plasma and mucus. And transferrin and lactobacteria completely binds iron in competitively binds iron in blood tissues and milk, thereby preventing its availability to microorganisms. So interferons also comprise a family of secreted alpha helical cytokines induced in response to specific external biomolecules of viruses or other pathogens through PLRs, and they can act in biocrine or autocrine models, as we know, for regulating innate and acquired immunity. There are different types of human interferon: alpha, beta, gamma, delta, omega, and virus infected cells produce interferon alpha and interferon beta. So they are host cell specific but not virus specific. Gamma interferon, also called as immune interferon, neutrophils, NK cells, macrophages. Interferons also result in resistance to viral replication. Induce enzymes to degrade viral mRNA, increase MHC1 expression. Okay. This is the pathway for virus infected cells. Interferons, they have been uninfected cells that induces enzymes that will be capable of degrading RNA, and they will be infected with the virus. Activation enzymes, degradation of viral cellular RNA, blocks viral and cellular protein synthesis. So inflammation is basically an attempt by the body to eliminate the non-noxious enzyme and restore and maintain homeostasis. Okay, so it's basically a fight that your body is giving. Okay, it's caused by invading organisms in the second line of defense. So the principles of inflammation can be an increase in blood supply, increase in vascular permeability to large cell molecules, enhanced migration of leukocytes across the local vascular endothelium, and reduction of size of inflammation. Inflammatory response characterized by redness, heat, swelling, and pain. The good side is it isolates the damaged area. It mobilizes effector cells and molecules to the side, and in the last stages, promoting healing. The bad side will be we don't know exactly what we're dealing with. There could be allergies, autoimmune disorders. Phagocytosis we have talked. It's the ingestion of microorganisms or particulate matter by cell. It's performed by phagocytes, certain types of white blood cells and derivatives of them. So they can be either macrophage monocyte, those are slow, but can phagocytose repeatedly, contain bactericidal enzyme, long-lived. Selected cells have ability to present silver. I think it's a neutrophils rapid phagocytosis but cannot phagocytose immediately as granules which contain bactericidal enzymes. It is short-lived and no ability to present antigen. So what are the mechanisms of phagocytosis? Microbes adhere to the phagocyte. The phagocyte forms pseudopods that eventually engulf the particle, bacteria, whatever, and the phagocytic vesicles containing some antigen, phagosome, which gets fused with the lysosome to call the phagolysosome, and microbes in fused vesicle is killed and digested by the lysosomal enzymes within the phagolysosome, leaving a residual body which is indigestible to digest, and residual matter is removed by exocytosis. Phagolysosome is a fusion of phagosome with lysosome. Granules of phagocyte take place by help of cytoskeleton, followed by the release of digestive and degradative enzymes. So following the attachment and polymerization and depolymerization of actin molecules, it sends pseudopods out to engulf the bacteria. That's how phagocytosis happens. Following engulfment, the bacterium is placed in a vesicle called a phagosome. Lysosomes move along the cytoskeleton and fuse with phagosomes to create phagolysosomes. Lysosome is digestive enzymes and microbicidal chemicals first with the phagosome containing the ingested bacteria to form a phagolysosome, and the bacteria is killed. Now we have oxygen dependent and oxygen independent system. So oxygen converted to superoxide anion, hydrogen peroxide, activated oxygen, etc. Whereas in case of oxygen independent system, we have the directional killing by lysozyme, lactoferrin, low pH cationic proteins, and hydrophilic and hydrophilic enzymes. So cytokines can also induce phagocytic cells, particularly macrophages, to produce nitric oxide as well, which can be toxic to microorganisms. So how nitric oxide dependent killing happens? It is inhibition of viral RNA. So this is inhibition of viral protein accumulation, inhibition of virus release from infected cell. Now, if the phagocyte is overwhelmed with microorganisms, the phagocyte will empty the contents of its lysosome by a process called degranulation in order to kill the microorganisms or cell separate extracellularly. These released lysosomal contents, however, also kill surrounding host cells and tissues, and most tissues destruction associated with these infections is a result of the process. Generally, both cells and bacteria that are suspended in body fluids have negative charges. Therefore, they tend to repel each other. Opsonins provide positive charges, and coating with opsonins promotes phagocytosis. And activated macrophages and other leukocytes, they can release pro-inflammatory cytokines like TNF alpha, IL-6, IL-1, etc. And the cytokines stimulate the anterior hypothalamus of the brain to produce prostaglandins that lead to an increase in body temperature. Then fever increases the physiological temperature above the optimum growth temperature for many microorganisms, right? And fever leads to the production of heat shock proteins, resulting in the production of inflammation promoting cytokines. So fever elevates the temperature of the body, decreasing the rate of the enzyme reactions, like and speeding up metabolism within the body. So what can be the pathogens? There are kind of different pathogens, bacteria, viruses, fungi, yeast. So antipathogen immunity comprises of an early innate response followed by a more sustained adaptive response. So all types of pathogens, mechanisms of innate immunity offer an immediate response and either prevents the establishment of infection or slows the infection down until adaptive immune mechanisms are induced. The components of simple innate immunity include various physical, chemical, and cellular factors such as epithelial layers, mucosal, antibiotic peptides, etc. And recognition of pathogens or their components by membrane-bound or soluble pattern receptors like TLRs induces inflammation, cytokine release, and complement activation. So, if above innate responses are insufficient to eliminate the invaders, adaptive immunity takes over. So, the DCs, or the dendritic cells and the macrophages, are key cells which act as the bridge between the innate and the adaptive immunity. And they initiate the latter by secreting cytokines. Cytokine that's produced will direct the induction of acquired immunity by triggering appropriate T or B cell responses. So we have almost come to the end of the first question. To discuss this. So coming back, we will not discuss in very great details about the immune response against other organisms. Coming back to the answer for today, degradation of pathogenic bacteria inside a host cell occurs in secretory vesicle, phagolysosome, SCR, smooth endoplasmic reticulum, RER, rough endoplasmic reticulum. Answer will be phagolysosome. That's correct. If any of you have any doubt, you can ask me now, or we can move on to the next question in short time. I'll stop for 2-3 minutes, then we move on to the next question. Okay, so we'll move to the next question for today. If there's no doubt regarding this question. Okay, so the lumen of lysosome has a pH of A 9 to 10, B 4 to 5, C 7 to 7.5, and D 8 to 9. So I'll again wait for 2-3 minutes. If you know the answer, let me know. We'll move on to the answer then.
Ma'am, the answer is option B.
4 to 5, right? Okay, so you're saying the lysosomal pH will be 4 to 5. So, yes, lysosome has an acidic pH, right? So, as we know that through chemical analysis of living things is possible because of its complex organization, the interaction of molecules and life in the form of living cell cannot be produced. And cell is the smallest living entity, smallest functional unit which serves as a building block for the immensely complicated whole body. Okay. So if you see life is the form of cell that cannot be produced. Like we can replicate cells. Like cells will be produced from cells only, right? So it's the smallest structural and functional unit. It can carry out living processes, functional activities related to the specific structural property. It can help build. Basically, these are living building blocks of animal or plant. And then organism structures and functions also depends upon characteristics of the cells. All new life and new cells are formed from pre-existing cells. Cells of one organism are fundamentally very similar in structure and function. Cells of a particular tissue of our body are also very fundamentally close, like similar in structure and function. Now they cannot be seen by naked eye, obviously they can be seen by microscope. So the plasma membrane encloses a cell. Then there are this is a nucleus which contains genetic material. And then cytoplasm, that's a portion of the cell interior not occupied by the nucleus. The plasma membrane is a thin membranous structure enclosing each cell. It has an oily barrier that holds contents of cell. It's a gated wall, selective movement of molecules. Nucleus is largest single organized compartment, spherical or oval near the center, surrounded by double layer. Nuclear membrane can have nuclear pores, allowing traffic between nucleus and cytoplasm. Genetic material is DNA. Now, what is the function of nucleus? Direct synthesis of proteins serves a genetic blueprint of during cell duplication, and structural enzymes controlling chemical reactions. They continue identical type of cells line within the body and in the reproductive cell to transfer genetic material. Okay. Now we have messenger RNA, ribosomal RNA, and transfer RNA. The arrows have gone all bad. Cytoplasm can be cytosol and organelles. Cytosol can be complex gel-like liquid site for compatible chemical reactions or elaborate protein network cytoskeleton. Gives shape, provides internal organization, and regulates its movements. Organelles can be distinct, highly organized, membrane enclosed, occupies about half total cell volume, separate compartments, separate contents, etc. 6 main types of organelles are there: endoplasmic reticulum, rough ER, smooth ER, Golgi complex, lysosomes, peroxisomes, mitochondria. It's really vacuoles. Okay. Vacuoles. Similar in cells that contain specific set of chemicals required for particular cellular function can carry out incompatible chemical reactions simultaneously as well. The endoplasmic reticulum, protein and lipid manufacturing factory, elaborate fluid filled extensively distributed membranous system. It's two types continuous with each other and the relative amount varies with the function of cell. We have talked about different kinds of endoplasmic reticulum, the SER, which is network of tiny interconnected tubules, the RER, which is projects outwards from SER as stacks of flattened sacs, right? Outer surface of the membrane studded with ribosomes, rough granular appearance. Now, new protein and ribosomal RNA, if it's released in the ER lumen, it is excreted as hormones or enzymes, construction of new cell membranes or organelles. Synthesis of lipids by enzymes present in the membrane, released to lumen with protein, and then pressed attachment of carbohydrate birth off as a transport vesicle. As I said, the smooth ER is called smooth ER because there is no ribosome attached to the ER, right? So that way this no ribosomes are not involved in protein synthesis, but it is involved in lipid synthesis. Smooth ER. It serves a central packaging and discharge site for molecules which are to be transported from ER. Formation of transport vesicles which contain new proteins and lipids and is membrane bound and passes to Golgi complex. Formation of peroxisomes and membrane use is replaced by newly formed protein and lipid. Okay, so there are different additional responsibilities of different cells as well. Like steroid secreting cells have abundant smooth endoplasmic reticulum. Liver cells are membranes of ACR contain enzymes involved in detoxification. In muscle cells, ACR stores calciums, which plays important role in process of muscle contraction. What is Golgi complex? There are stacks of flattened, curved, membrane-bound sacs or cisterns. They may not be physically connected with each other. They are thin at the center and dilated at the periphery, and the number varies. Cells specialized in photosynthesis, we have hundreds of sacs. Now, what is the mechanism of function? Transport vesicles containing cargo from smooth endoplasmic reticulum fuses with the innermost sac of Golgi complex. As material travels through the layers of sacs to the outer sacs in the form of transport vesicles. During transit, the raw material will be converted to final finished product. Sorting and directing the finished product, secretion of exterior of cell, tissue to the exterior of cell, construction of new plasma membrane and incorporated in other organelles like lysosomes. Now, membrane is specific proteins recognition marker for cargo on the inner surface. Then there will be core proteins for curling membrane and then docking on the outer side of the inside core protein, like the snare proteins. If you have read about the snare proteins. Now, what is the cargo? Concentration of finished product with appropriate amino acid sequence acting as a sorting signal. Now, what is the process of exocytosis? Binding of vesicles in cytosol, separating specialized finished products from cytosol. Then they'll move towards the membranes with appropriate signals. Attachment with special protein markers, target membrane like snares, then fusion of membrane, opening of vesicle, and release of secretion. Now, what are the peculiarities of the secretory process? So once protein is synthesized, does not come in contact with cytosol. Synthesis and storage are well ahead of time of requirement, so different secretory vesicles for different destinations. Then we have the lysosomes, a membrane enclosed sacs containing powerful hydrolytic enzymes. Average number will be 300 per cell. There is no fixed structure, vary in size and shape, 0.2 to 0.5 micron in diameter. Then they are granular when inactive. Then membrane protects the rest of the cell. Then membrane and enzymes from Golgi complex. Extracellular material to be tackled by lysosome is brought into the cells by endocytosis. There can be three types: pinocytosis that happens in all cells, receptor mediated endocytosis, and then phagocytosis. Pinocytosis is cell-breaking, non-selective, continuous process seen in all cells. Membrane deforming protein attached to membrane. Formation of pouch by dipping of the membrane. Then the ends are sealed. Endocytic vesicle with ECF. Then vesicle is pinched off by protein dynamin. Okay. So that is how the pinocytosis process occurs. You see, this is how it occurs. This is a schematic. What is receptor-mediated endocytosis? Basically, highly selective process to import important specific large molecules requires energy and calcium. So if you see, there is a coated pit formed of clathrin, actin, myosin. Though the substance is taken in like this. Then we have phagocytosis, which is internalization of large multimolecular particles by specialized cells such as certain types of WBCs. This is phagocytosis. Internalization, now phagosome. Okay. Now, what is autophagy? The role in regression of organ, uterus, mammary glands. So, it basically is the removal of aged or damaged organelles. Rupture of lysosomal membrane causes cell destruction, but minimal damage because optimum pH for hydrolytic enzymes is acidic. Damage to nuclear DNA alters genetic properties. So, deficiency of one or more enzymes can lead to storage diseases like Tay-Sachs disease. We have talked about peroxisomes. There are several hundreds. They help with transport of H plus across membranes so acidic pH. Membrane enclaves sacs with powerful oxidizing enzymes which use oxygen to remove hydrogen from organic molecules and detoxify waste produced in the cells or foreign toxic compounds by formation of H2O2, which is oxidant but accumulation is prevented by catalase, which is antioxidant. The mitochondria, where the electron transport chain happens, the outer membrane, inner membrane, intermembrane space where the chemiosmotic reactions happen, and the matrix where the dissolved enzymes for citric acid cycle are there. So there are every organelle are present in every cell. Extract energy from nutrient and transfer to usable form, and they possess their own DNA, mitochondria, which is why it produces molecules required for generating energy. Defect in DNA can lead to degenerative disease or aging, and double membrane, basically outer smooth and inner folded membrane forming cristae, basically to increase the surface area. Synthesis of ATP is oxidative phosphorylation, release of energy due to electron transport reactions used for active uptake of hydrogen by inner membrane, and the accumulation of these hydrogens in the intermembrane space, transport of these hydrogen ions through channels in inner membrane, adaptation of ATP synthetase attached to the channel protein, etc. What is chemiosmotic reactions? We have already discussed about all of this in the last class, so I will not be going into huge detail. Only the uses of ATP, basically synthesis of new chemical compounds for secretion and growth, membrane transport, mechanical work. Cytosol. Cytosol is semi-liquid surroundings organelles, highly organized gel-like mass with different composition of different sites. Cytoskeleton is disposed throughout enzymes regulating intermediary reactions, obviously ribosomal protein synthesis, storage of fats, etc. They can be differentiated into intermediate filaments, microfilament, and microtubules. Now, microtubules are the largest skeletal element, slender, long, and hollow unbranched tubes. Functions are obviously maintaining asymmetric cell shape, like in axon, transport of secretory vesicles and other materials in any direction by use of motor protein and energy, movement of specialized cell projections such as ciliary flagellum, and distribution of chromosomes during mitosis of spindles. And we have the microfilament, smallest element of cytoskeleton. Actin is present in most cells, and two strands of globular actin are there. Their roles in the cell are cellular contractile system, mechanical stiffener for cellular projection, and microvilli. Okay. There are intermediate filaments like TUF. They maintain structural integrity of cell and resist mechanical stress. Microfilaments in axon, keratin in skin cells. The functional systems of cells are ingestion, digestion of foreign substances, synthesis and formation of new structures, energy extraction, and locomotion. Coming to the vesicles, the cellular organelles cell is composed of a lipid bilayer, and they also function in metabolism and enzyme storage as well. They can be vacuoles, lysosomes, transport vesicles. and secondary vesicles. So for vacuoles, vesicles that contain mostly water, they're able to regulate the pressure and water level of the cell to control the conditions of the internal environment. This is an image of vacuoles. Lysosomes are basically spherical vesicles which contain hydrolytic enzymes that can break down virtually all kinds of biomolecules. As you can see, the lumen's pH is 4.5 to 5, which is very acidic, and it is optimal for enzymes involved in acidic hydrolysis. Besides degradation of polymers, the lysosome is also involved in various cell processes, including secretion, plasma membrane repair, cell signaling, and energy metabolism. Lysosomes can also act as the waste disposal system of the cell by digesting unwanted materials in cytoplasm. Lysosomes are known to contain more than 60 different enzymes. So enzymes of the lysosome are synthesized in the rough endoplasmic reticulum, and the enzymes are imported from the Golgi apparatus in small vesicles which fuse with larger acidic vesicles. We have centrioles, which are basically animal cells, all animal cells have these two small organelles. The centrioles can help in the cells to divide. They are seen in the process of mitosis and meiosis. Centrioles together are typically located near the nucleus in the centrosome. And centrioles are made up of 9 bundles of microtubules that are arranged in a ring. And the absence of centrioles causes divisional errors and delays in the mitotic process. Centrioles, the nucleus, which basically acts like the brain of the cell. Not all cells have a nucleus. This prokaryotic, eukaryotic. Prokaryotic is nuclear, eukaryotic is nucleus. It has two major functions. Basically, it stores the cell's hereditary material or DNA, and it coordinates the cell's activities, which include growth, intermediary metabolism, protein synthesis, reproduction, etc. So the Golgi apparatus plays a central role in vesicular traffic within cells, the post office of the eukaryotic cell. That is what it is. So this image has been taken from the molecular biology of the cell book. So this is how all the pathways happen. So you can see the different color: blue, red, green. Now proteins that reside in the ER lumen can escape in a transport vesicle, right? They are captured by the empty KDEL receptors that bind a KDEL sequence like lysine, asparagine, glutamine, and leucine. Okay. On ER proteins. So what is this KDEL sequence? So this KDEL sequence is the target peptide sequence in mammals and plants, basically located on the C-terminal end of the amino acid structure of a protein, right? So the K stands for lysine, D is for aspartic acid, sorry, not asparagine, aspartic acid, E is glutamine and glutamic acid, sorry, and L is leucine. That is how the KDEL sequence happens. Soluble resident protein will basically remain in the ER as long as it contains a KDEL sequence on the C-terminal end of the protein. Now, since vesicle budding is such a dynamic process and has a high concentration of soluble proteins in the ER, soluble proteins are inadvertently transported to the cis Golgi via coated vesicles, and the transportation mechanism of proteins containing the KDEL sequence is facilitated by KDEL receptors attached to COP2 COP2 and COP1 receptors. If you can see over here, this is the retrieval pathway, this is the forward pathway. So the Golgi apparatus is also an ordered series of compartments. We have the cis phase and the trans phase. So we don't need to go into much detail about all of this. So lysosomes are sites of intracellular digestion. Okay, the cytosol pH is 7.2. Lysosomal pH will be 4.5 to 5. It's acidic. So there are high acid hydrolysis, nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases, etc. This is also images taken from the molecular biology of the cell book 4th edition. This is a lysosomal hydrolase, end-linked oil cyanide with terminal mannose residue, and this is a signal patch. The binding of the signal patch to the recognition site, and then transfer of the GLC-NAC phosphate to mannose in the catalytic site. This will be the Glicnac phosphotransferase. Then transfer of this Glicnac phosphate to mannose in the catalytic site, and then release. So the Glicnac is then clipped off, leaving mannose 6 phosphate. So the lysosomal hydrolase with Glicnac phosphate attached to mannose in oligosaccharides. That's what is shown here. And transfer of lysosomal enzymes to late endosomes, which become the lysosomes. That is also what is shown over here. Receptor cycling, receptor dependent transport binding to mannose 6 phosphate receptor. So coming back to the question that we started with, the lumen of lysosome does have a pH of 4 to 5, 4.5 to 5. Okay. Anybody having any question and doubt in this question? Then ask me. We'll wait for 2-3 minutes and then move on to the next question for today. Okay, so we move on to the next question. Which of the following is a cofactor of hexokinase? Fe2 plus iron, CO2 plus cobalt, Ca2 plus calcium, Mg2 plus magnesium. Anybody?
I think Mg2 plus.
Okay. Anybody has a different answer? So yes, it's Mg2 plus. So we'll start with a little bit about enzymes and why do they need cofactors. So biomolecular is the protein or RNA that can catalyze a specific chemical reaction, that is enhance or speed up the reaction rate of reaction in providing a reaction path with the lower activation energy can be called an enzyme. So the catalytic power speeding can up reactions to 10 to the 8, 10 to the power 20 fold even. Okay, but they do not get used up themselves. Another property will be specificity. Some for substrate can range from absolute to relative for the reaction to be catalyzed, and some enzymes will be regulated. That means they can sense metabolic signals and they can itself also be regulated. Now, the catalytic power is defined as the ratio of the enzyme catalyzed rate of the reaction to the uncatalyzed rate. For example, urease at 20 degrees Celsius, the rate constant for the enzyme catalyzed reaction is 3 into 10 to the power 4 per second. Right? The rate constant for the uncatalyzed hydrolysis of urea is 3 to 10 to the power 10 seconds per second. Right? And 10 to the power 14 is the ratio of the catalyzed rate to the uncatalyzed rate of the reaction. Now, specificity, now defined as a selectivity of enzymes for the reactants upon which they act. In an enzyme catalyzed reaction, none of the substrate is diverted into non-productive side, so no wasteful byproducts are produced. Substances upon which enzymes are traditionally called substrates, the selective qualities of an enzyme are collectively recognized. Specificity and the specific site of the enzyme where substrate binds and catalysis occurs is called the active site. Then we have regulation. Now, regulation of enzyme activity ensures that the rate of metabolic reactions is appropriate to cellular requirements. It is essential to the integration and regulation of metabolism. It can be achieved by various ways: inhibitor, activator, hormonal, and rate of synthesis. Right? So we will not go into classification. So they are often the enzymes are often named by adding the suffix ase to the name of the substrate upon which they acted. So the names bearing little resemblance to their activity, like catalase, peroxide decomposing enzyme. So if you see, there are different so glucose phosphotransferase is class name is transferase, subclass is phosphotransferase, subclass is hydroxyl group as a acceptor, specific enzyme is D-glucose is phosphoryl group acceptor. Enzyme classification, there are 6 classes. Oxidoreductase, so transfer of reducing equivalence from one redox system to another. Like alcohol dehydrogenase, lactate dehydrogenase, cytochrome oxidase. Then there are transferases, like functional group is transferred from one compound to another, like kinases. Then hydrolase, like cleave CO, CN, CS or PO, etc. bonds by adding water across a bond, like lipase, acid phosphatase. In lyase, like cleave CO, CN or CS bonds, but do so without addition of water and without oxidizing or reducing substrates. Hydrolase, fumarase, carbonic anhydrase. Then there will be isomerases. Basically, what they do is they catalyze intermolecular rearrangements of functional groups that reversibly interconvert to optical or geometric isomers like triose isomerase, phosphoexose isomerase, mutase, etc. Then we have ligase, basically which catalyze biosynthetic reactions that form a covalent bond between two substrates, utilizing ATP-ADP interconversion. Glutamine synthetase, DNA ligase. The specificity means highly specific compared to other catalysts. It catalyzes only specific reaction. Now, 3 types of reactions: stereo specificity, optical specificity, reactant specificity, and substrate specificity. Okay. Optical specificity is basically able to recognize optical isomers of the substrate. They can act only on one isomer. The enzymes of the amino acid metabolism, D and L amino acid oxidase. Isomerases do not exhibit stereospecificity. When we have reaction specificity, they catalyze only one specific reaction over substrate. Amino acid can undergo deamination, transamination, decarboxylation, and each is catalyzed by a separate enzyme. Now, specific towards the substrate will be called substrate specificity, like glucokinase, galactokinase, both transfer phosphoryl group from ATP to different molecules. There can be three types: absolute, relative, substrate, and broad. Absolute substrate specificity means that they act only on one substrate, like urease. Relative specific substrate specificity means they act on structurally related subclasses. Further divided into group dependent, like they act on specific groups like trypsin, break peptide bond between lysine and arginine, chymotrypsin, can act on aromatic amino acids. And then bond specificity, like act on specific bond like proteolytic enzyme, glycosylase, etc. Bond specificity is act on closely related substrates like hexokinase act on many hexoses. Okay. Now the protein or RNA can be catalytic, like catalyst. Okay, so they can be RNA. They can be enzymes. So tertiary structural specific conformation are very essential for catalytic power because that will determine the active site, right? So holoenzyme will be the functional unit that is called the holoenzyme, and apoenzyme coenzyme will discuss. So prosthetic group, if we add to it, that is a non-protein molecule. Coenzyme and cofactor is also non-protein molecule. Prosthetic group is however very tightly covalently bound, whereas coenzyme cofactor are loosely bound. Prosthetic group is like stable incorporation. Coenzyme cofactor are dissociable. And prosthetic group will be like they cannot be dissociated. Coenzyme or cofactors can be separated by dialysis. Now there can be monomeric enzymes like made up of a single polypeptide like ribonuclease, trypsin. Oligomeric enzymes are more than one polypeptide like LDH, lactate dehydrogenase, aspartate carbamylase. Multi-enzyme complexes will be specific sites to catalyze different reactions in sequence. Only native conformation is active, not individual, like pyruvate dehydrogenase. And in a number of metabolic pathways, several enzymes which catalyze different stages of the process have been found to be associated non-covalently, giving a multi-enzyme complex, like pyruvate dehydrogenase complex in electron respiratory chain. In other cases, different activities may be found on a single multifunctional polypeptide chain. The presence of multiple activities on a single polypeptide chain is usually the result of a gene fusion event. Okay. Now we know about the general characteristics of enzymes, like usually a protein acting as a catalyst in specific biochemical reactions. Each cell in the human body contains thousands of different enzymes. Every reaction in the cell requires its own specific enzymes. Most enzymes are globular proteins, like a few enzymes are made up of RNA and catalyze biochemical reactions involving nucleic acids. Enzymes undergo all reactions of proteins. Enzymes denaturation due to pH or temperature change. A person suffering with high fever will run the risk of denaturing certain enzymes, right? So simple enzymes are composed of only protein. Conjugated enzymes are composed of apoenzyme, conjugate enzyme without its cofactor, and the protein part of the conjugated enzyme. Coenzyme will be the non-protein part of a conjugated enzyme. So the apoenzyme can't catalyze its reaction without its cofactor. So the combination of the apoenzyme with the cofactor makes the conjugated enzyme functional. So holoenzyme is like apoenzyme plus cofactor. It is a biochemically active conjugated enzyme. So coenzymes provide additional chemically reactive functional groups besides those present in the amino acids. So the apoenzymes, they are either small organic molecules or inorganic ions. So metal ions often act as additional cofactors like zinc, magnesium, manganese, iron. So metal ion cofactor can be bound directly to the enzyme or to a coenzyme. A coenzyme will be a small organic molecule acting as a cofactor in a conjugated enzyme, as I said. And coenzymes are derived from vitamins or vitamin derivatives. So many vitamins act as coenzymes like B vitamins. B vitamins. So enzyme is something that can facilitate a chemical reaction. Coenzyme is a compound derived from a vitamin like NAD+, nicotinamide adenine, that assists an enzyme in facilitating a chemical reaction. Cofactor is a metal ion like magnesium 2 plus that assists an enzyme in facilitating a chemical reaction. Apoenzyme is a protein only part of the enzyme like isocitrate dehydrogenase. that requires an additional coenzyme to facilitate the chemical reaction. Holoenzyme, combination of the apoenzyme and coenzyme which together facilitate a chemical reaction. Enzyme nomenclature. Enzymes are named according to the type of reaction they catalyze or the substrate. Substrate is the reactant upon which the specific enzyme acts, like enzyme physically binds to the substrate. Like lactase, amylase, lipase, or protease, suffix of an enzyme can be ase. It denotes an enzyme. So digestive enzymes have the suffix in, like pepsin, trypsin, and chymotrypsin. Prefix denotes the type of reaction the enzyme catalyzes, like oxidase is a redox reaction. Hydrolase is the addition of water to break one component into two parts. Then we have substrate identity. It's often used together with the reaction type. Like pyruvate, carboxylase, lactate, dehydrogenase. There are 6 major classes of enzymes based on the type of the reaction they catalyze. Okay. Oxidoreductase will be redox reaction, reduction and oxidation. Examples are dehydrogenases, which catalyze reactions in which the substrate is either oxidized or reduced. Substrate is oxidized and reduced. Transferase will be transferring a functional group from one molecule to another, like transaminases, which catalyze the transfer of amino group, or kinases, which catalyze the transfer of phosphate groups. Hydrolysis reaction. It lipases catalyze the hydrolysis of lipids, and proteases catalyze the hydrolysis of proteins. Then lyase, addition removal of atoms to form a double bond. Decarboxylases catalyze the removal of carboxyl groups. Isomerization reaction. The isomerases may catalyze the conversion of an aldolase to a ketose, and mutases transfer functional group from one atom to another within a substrate. Then ligase, synthetic synthesis reaction, joining two molecules into one, forming a new chemical bond, coupled with ATP hydrolysis. Synthetases link two small molecules to form a larger one. Now there is an enzyme active site, the specific portion of an enzyme where the substrate binds, where it undergoes a chemical reaction. This active site is a 3D-like crevice-like cavity formed by secondary and tertiary structures of the protein part of the enzyme. So an enzyme can have more than only one active site. The amino acids or R groups in the active site are important for determining the specificity of the substrate. So when the substrate binds to the enzyme active site, an enzyme substrate complex is formed temporarily. It allows the substrate to undergo its chemical reaction much faster. Right? If you see the images, the active site, this is an enzyme which is an active site, then substrate. The active and site and substrate both change shape a little bit, and you form the enzyme substrate complex, right? And then the enzyme product complex and products. And then we have this is the substrate. Here, this is the active site. It changes. This basically talks like the lock and key model. This basically is more similar to the induced fit model. So the lock and key model of enzyme action is the active site is fixed, if you see over here, with a rigid shape like a lock. Substrate key must be fit exactly into the rigid enzyme lock. So complementary shape and geometry between enzyme and substrate, the key fits into the lock. Now upon completion of the chemical reaction, the products are released from the active site, so the next substrate molecule can bind. Whereas induced rate of enzyme reaction, so many enzymes are flexible and constantly change their shape. The shape of the active site changes to accept and accommodate the substrate. So the conformation changes in the enzyme's active site to allow the substrate to bind. So the analogy, a glove or an enzyme changes shape when a hand is inserted into it. Now we have absolute specificity. So an enzyme will catalyze a particular reaction for only one substrate. Most restrictive of all specificities, so not common. Catalase has the absolute specificity for hydrogen peroxide. Urease catalyzes only the hydrolysis of urea. Now group specificity, the enzyme will act only on similar substrates that have a specific functional group. Like carboxylpeptidase cleaves amino acids one at a time from the carboxyl end of the peptide chain. Hexokinase adds a phosphate group to hexoses. Then linkage specificity. So the enzyme will act on a particular type of chemical bond irrespective of the rest of the molecular structure. So the most general of the enzyme specificity is phosphatases. The hydrolyze phosphate ester bonds in all types of phosphate esters. Then chymotrypsin catalyzes the hydrolysis of peptide bonds. Stereochemical specificity, the enzyme can distinguish between stereoisomers. So chirality is inherent in an active site. So L-amino acid oxidase catalyzes reactions of L-amino acids but not of D-amino acids. Then what are the factors affecting enzyme activity? Measure the enzyme activity is basically the rate at which an enzyme converts substrate to products in a biochemical reaction. Four factors are affect enzyme activity like temperature, pH, substrate concentration, and enzyme concentration. Right. So with increased temperature, the energy in the kinetic sense will increase. So there will be more collisions, increased reaction rate. So there is an optimum temperature at which the enzyme exhibits maximum activity. For human enzymes, it is 37 degrees Celsius because that is the physiological temperature. When the T increases beyond the optimum temperature, the changes in the enzyme's tertiary structure will occur. Basically, the enzyme might get denatured, the inactivated, and little activity will be seen after that. So reaction rate again goes down. For pH, it's similar. There is an optimum pH at which the enzyme exhibits maximum activity. Most enzymes are active over a very narrow pH. The protein and amino acids are properly maintained. Small change in pH can result in enzyme denaturation and loss of function. So each enzyme has its characteristic pH optimum, which usually falls within the physiological pH. Like pepsin is in stomach, so its outer pH will be very acidic, like 2, 3. Trypsin will be around 8. Now, substrate concentration. If enzyme is kept constant and the substrate is increased, the reaction rate increases until a saturation point is met. At the saturation point, the reaction stays the same even if substrate is increased. This is basically the Michaelis Menten reaction equation. So, at saturation point, substrate molecules are bound to available active sites of the enzyme molecules. Reaction takes place at the active site. If they are all active sites occupied, the reaction is going at its maximum rate. Each enzyme molecule is working at its maximum capacity. Incoming substrate molecules must then wait their turn. Now, if the substrate is kept constant and the enzyme is increased, the reaction rate increases. So, the greater the enzyme, the greater the reaction rate. So, the rate of an enzyme catalyzed reaction is always directly proportional to the amount of the enzyme present. So, in a living cell, the substrate is much higher than this enzyme. So, enzymes are not consumed in the reaction. Enzymes can be reused many times. Okay. So formation of an enzyme substrate complex as an intermediate species provides an alternative pathway with lower activation energy through which a reaction can occur. Either the lock and key model, the active site is a fixed geometric shape, only substrate with a matching shape can fit into it, or the induced fit model. The active site is a flexible shape that can change to accept a variety of related substrates. So enzymes vary in their degree of specificity for substrates. And these are different factors that can affect the rate of the enzyme activity. So the reaction rate will increase with increasing enzyme concentration as well, assuming enzyme concentration is much lower than that of the substrate. Okay. So there are different inhibitors also in case of if you want to stop the action of enzymes. Okay, it can be a substance that can slow down or stop the normal catalytic function of an enzyme by binding to the enzyme. Reversible competitive inhibition, reversible non-competitive inhibition, irreversible inhibition. Okay, we'll not go into too much detail. Wait, little bit. Reversible competitive inhibition. So competitive inhibitor will resemble the substrate. If you see like this. So inhibitor basically competes with the substrate for binding to the active site of the enzyme. So if an inhibitor is bound to the active site, the substrate can no longer bind to it because see it basically resembles the substrate. So it prevents substrates from getting access to the active site. So it basically decreases or stops the enzyme activity. Now binding of the competitive inhibitor to the active site is a reversible process. So if you add many more substrates to outcompete the competitive inhibitor, then the substrates will go and bind. Like antihistamines are inhibit histidine decarboxylase, which converts histidine to histamine. Then we have reversible non-competitive inhibition. So a non-competitive inhibitor decreases enzyme activity by binding to a site on the enzyme other than the active site. So it basically binds to another site and changes the tertiary structure of the enzyme and alters the active site. So the substrate no longer can bind to it and it decreases enzyme activity. This process can also be reversed by lowering the non-competitive inhibitor. Like heavy metals like Pb and Hg bind to the SH of domain of the cysteine away from the active site. So it disrupts the secondary and tertiary structure. Then we have irreversible inhibition. It basically inactivates an enzyme by binding to its active site by a strong covalent bond. Okay, so it permanently deactivates the enzyme. So that molecule of enzyme cannot function anymore. So they do not resemble the substrates. So addition of excess substrates cannot reverse the reaction because it is irreversible. So that is the basic uh, details of these different types of inhibitions. Then we have allosteric enzymes. Allosteric enzymes have a quaternary structure. They are composed of two or more protein chains. They possess two or more binding sites. So what happens? One binding site is for substrate active site, and one binding site is for the regulatory molecule regulatory site. So active and regulatory binding sites are distinct from each other in shape and location. One second. One second. This slide got. So binding of a regulatory molecule to its regulatory site causes changes in the 3D structure, tertiary structure of the enzyme and the active site, right? So the binding of the positive regulator will upregulate enzyme activity, which will make enzymes enhance the active site more able to accept the substrate. Binding of any kind of negative regulator will downregulate the enzyme activity. It will compromise the active site and it will be less able to accept the substrate. It will not go into much details. We'll talk about this some other day. So if we talk about glycolysis, that's where hexokinase comes from. So glycolysis basically is one molecule of glucose is degraded into two molecules of pyruvate. Like 6 carbon molecule is degraded into 2 3 carbon molecules, and the free energy released in this process is stored as 2 molecules of ATP and 2 molecules of NADH. Right? So in standard conditions, glycolysis is an hexagonic reaction. Okay. Now it is also known as the Embden-Meyerhof-Parnas pathway. It is defined as the sequence of reactions converting glucose or glycogen to pyruvate or lactate with production of ATP, and enzymes take place in cytosomal fraction of the cell. A major pathway in tissues lacking mitochondria like erythrocytes, cornea, lens, and it is essential for brain, which is dependent on the glucose for energy. Okay, brain is totally dependent on glucose for energy. Under anaerobic condition, glucose is not converted into pyruvate. Rather, glucose is converted into lactate fermentation process. So if you see the weight of glucose in living systems, 5.2% of total free energy that can be released by glucose is released in glycolysis. Now, glycolysis was the very first biochemistry or oldest biochemistry study. It is the first metabolic pathway discovered, and fermentation is caused by microorganisms. Pasteur's effect. The aerobic growth requires less glucose in anaerobic condition. Then Buchner stated that reactions of glycolysis can be carried out in cell-free yeast extract. Hardin and Young said that inorganic phosphate is required for fermentation. Yeast extract could be separated in small molecular weight essential Coenzymes are what they call cozymes, and bigger molecules called enzymes or zymases. With the efforts of many workers, complete pathways for glycolysis was established. If you see, glucose will undergo glycolysis, then successive reactions to form 2 pyruvate. Under anaerobic conditions, it can give rise to 2 ethanol plus 2 CO2 fermentation to alcohol and yeast, and also 2 lactate, like fermentation to lactate in vigorously contracting muscle, erythrocytes, or other cells, and in some microorganisms. So when you run for a long time on your or you're climbing stairs, you start panting and your legs calf muscles start paining, right? That's because of lactate accumulation. Or pyruvate under aerobic conditions, pyruvate will get converted to acetyl CoA, which will then go into Krebs cycle or the TCA cycle, tricarboxylic acid cycle or citric acid cycle. Now there are 10 enzyme catalyzed reactions in glycolysis in 2 stages. Okay. So reactions 1 to 5 is a preparatory stage in which a glucose is phosphorylated, converted to fructose, which is again phosphorylated, and cleaved into 2 molecules of glyceraldehyde 3-phosphate. In this stage, there is investment of 2 molecules of ATP. In stage 2, the 2 molecules of glyceraldehyde 3-phosphate are converted into pyruvate with concomitant generation of 4 ATP molecules and 2 molecules of NADH. That there is a net gain of 2 ATP molecules per molecule of glucose in glycolysis. Okay. Now, what are the importance of phosphorylated intermediates? So, the possession of the negative charge which inhibit the diffusion through membrane is the conservation of free energy in high energy phosphate bond and then the facilitation of catalysis as well. In the preparatory phase, phosphorylation of glucose and its conversion to glyceraldehyde 3-phosphate, if you can see over there. Okay. This is the first primary reaction, then second primary reaction, then the cleavage of 6 carbon sugar phosphate to 3 carbon sugar phosphate. Then there is the payoff phase, which is basically oxidative conversion of glyceraldehyde 3-phosphate to pyruvate and the coupled formation of ATP and NADH. So the oxidation and phosphorylation happens. First ATP forming reaction, substrate level phosphorylation. Then second ATP forming reaction, which is again second substrate level phosphorylation. Now talking about hexokinase. Hexokinase helps in phosphorylation of hexoses, mainly glucose, right? Hexose like 6 carbon. Now this enzyme is present in most cells. In liver, glucokinase is the main hexokinase. They are isoenzymes which prefers glucose as substrate. It requires magnesium ATP complex as substrate. So magnesium is a cofactor. Uncomplexed ATP is a potent competitive inhibitor of this enzyme. So without magnesium, this ATP becomes inhibitor of the enzyme. But once you add magnesium, it acts as a cofactor and helps in the reaction. Enzyme catalyzes the reaction by proximity effect, bringing the two substrates in close proximity. And this enzyme undergoes large conformational change upon binding with glucose. It is inhibited allosterically by glucose 6-phosphate. Now, what are the differences between hexokinase and glucokinase? In case of hexokinase, the Km is low, high affinity for glucose, whereas the opposite is true for the other. So hexokinase is non-specific and phosphorylate any of hexoses, whereas glucokinase can only phosphorylate glucokinase. Hexokinase is not affected by insulin. Glucokinase is stimulated by glucose and insulin. Hexokinase can be allosterically inhibited by glucose, whereas it is not whereas glucokinase is not inhibited by glucose 6-phosphate. Okay. Then the next enzyme will be phosphoglucose isomerase or phosphohexose isomerase. Isomerization of 6 glucose 6-phosphate to fructose 6-phosphate. So this enzyme catalyzes a reversible isomerization of glucose 6-phosphate and aldohexose to fructose 6-phosphate ketohexose. This enzyme also requires magnesium for its activity. It's specific for glucose 6-phosphate and fructose 6-phosphate. Then we have the phosphofructokinase 1 reaction. Transfer of the phosphoryl group from ATP to C1 of fructose 6-phosphate to produce fructose 1,6 bisphosphate. So this step is an important irreversible regulatory step. The enzyme phosphofructokinase is one of the most complex regulatory enzymes with various allosteric inhibitors and activators. So ATP is an allosteric inhibitor and fructose 2,6 bisphosphate is an activator of this enzyme. So ADP and AMP, adenosine diphosphate and adenosine monophosphate also activate phosphofructokinase 1 activity, whereas citrate is an inhibitor. This step also requires Mg plus as a cofactor. If you can see the schematic over here. Then we have aldolase reaction, is cleavage of fructose 1,6 bisphosphate into glycerol 3 phosphate is an aldose, and dihydroxyacetone phosphate is a ketose. This enzyme catalyzes the cleavage of F1,6 bisphosphate by aldol condensation mechanism. So as shown here, the standard free energy change is positive in the forward reaction. Reaction concerning it requires energy. Since the product of this energy reactions are depleted very fast in the cells, this reaction is driven in forward direction by the later two reactions. Then you have the triose phosphate mutase reaction, conversion of dihydroxyacetone phosphate to glyceraldehyde 3 phosphate. So this is a reversible reaction catalyzed by acid-base catalysis in which histidine 95 and glutamate 165 of the enzymes are involved. You can see over there. Then the payoff phase starts where the glyceraldehyde 3 phosphate dehydrogenase reaction is conversion of GAPDH, GAP to base phosphoglycerate. So this is the first reaction of the energy yielding state. With the payoff stage, an oxidation of the aldehyde derives the formation of a high energy acyl phosphate derivative. So inorganic phosphate is incorporated in this reaction without any expense of ATP, and NAD plus is the cofactor in this reaction which acts as an oxidizing agent. So the free energy released in the oxidation reaction is used in the formation of acyl phosphate. The mechanism of this reaction is that iodine inhibits this reaction, indicating the involvement of cysteine residue of enzyme, and tritium from GAP is transferred to NAD, indicating transfer of hydride ion in oxidation reaction. 32P exchanges with phosphate, indicating acyl enzyme intermediate. So glyceraldehyde 3 phosphate binding to the enzyme is the first step. Then there is a nucleophilic attack by the SH group, sulfhydryl group, on the aldehyde group, forming a thiaminacetyl acetyl, and then direct transfer of the hydride to the NAD plus, leading to the formation of thioester. Energy of this oxidation is conserved in synthesis of thioester NAD. Okay. Another molecule of NAD plus replaces NADH from the enzyme side. Nucleophilic attack on thioester by phosphate to form 1,3-bisphosphoglycerate. Okay. So this is the reaction. Then there is a 7th step. Phosphoglycerate reaction, kinase reaction. So the transfer of phosphoryl group from 1,3-bisphosphoglycerate to ADP generating ADP. So this name of the enzyme indicates its function for reverse reactions. It characterizes the formation by proximity effect. ADP magnesium also needs magnesium at this step. Bind to the one domain and 1,3-BPG or bisphosphoglycerate binds to the other, and a conformational change brings them together similar to hexokinase. This reaction and the 6th step are coupled reaction generating ATP from the energy released by the oxidation of 3-phosphoglycerate. Okay, this step generates the substrate level phosphorylation because it generates ATP. Then we have the phosphoglycerate mutase reaction, conversion of 3-phosphoglycerate to 2-phosphoglycerate. Okay, so in active form, the phosphoglycerate mutase is phosphorylated at histidine 179. So there is transfer of the phosphoryl group from enzyme 2,3-PG, generating enzyme bound 2,3-bisphosphoglycerate intermediate, and this compound has been observed occasionally in reaction mixture. In the last step of the reaction, the phosphoryl group from the C3 of the intermediate is transferred to the enzyme and 2-PG is released. And in most cases, 2,3-BPG is present in trace amount, but in erythrocytes, it is present in significant amount, where it regulates oxygen affinity to hemoglobin. Then you have the enrolase reaction, dehydration of 2-phosphoglycerate to phosphoenol pyruvate. So dehydration of 2-phosphoglycerate by this reaction increases the standard free energy change of hydrolysis of phosphonitride bond, and rapid extraction of proton from C2 position by a general base on enzyme generating a carbanion, and the abstracted proton is readily exchanged with solvent. The The second rate limiting step involves elimination of the OH group, generating PEP, phosphoenol pyruvate. Second last step. And then the pyruvate kinase reaction, it transfers the phosphoryl group from PEP, phosphoenol pyruvate, to ADP, generating ATP and pyruvate. So this is second for substrate level phosphorylation of the reaction of glycolysis, and this enzyme couples the free energy of PEP hydrolysis to the synthesis of ATP. Second step of substrate level phosphorylation. This enzyme requires both Mg 2 plus and K plus potassium. Okay, so from one molecule of glucose, if you see, two molecules generated can directly be used for doing work on synthesis, and the two NADH molecules are oxidized in mitochondria under aerobic condition, and the free energy release is enough to synthesize 6 molecules of ATP by oxidative phosphorylation. So under the aerobic conditions, pyruvate is catabolized further in mitochondria through pyruvate dehydrogenase and citric acid cycle, where all the carbon atoms are oxidized to CO2, and the free energy is released is used in synthesis of ATP, NADH and FADH2. So under anaerobic condition, pyruvate is converted to lactate in homolactic fermentation and ethanol in alcoholic fermentation. So coming back to the question that we started with, which of the following is a cofactor of hexokinase? So obviously none of iron, cobalt or calcium are there. It's magnesium. Okay, so magnesium not only is a cofactor of hexokinase but many more enzymes of glycolysis, as we saw. Anybody has any question or a doubt regarding this question? If yes, then we can continue discussion. Otherwise, we can stop the session for today. Okay, so if there is no question, we can conclude the session today, and we'll meet again next Tuesday. Okay, we can leave the session. Thank you, everyone.
Thank you, ma'am. Thank you, ma'am.